Okay, so we're moving on to the next segment where we'll be discussing proteins, the second biomolecule, and the first of the real macronutrients that we're going to talk about, the others being carbohydrates and lipids or fats. So what is a protein? A protein is a polymer. We've been saying that these biopolymers, proteins are one of them. What is a polymer? A polymer is a large molecule or a macromolecule made up of many repeating subunits of smaller molecules, smaller what are called residues. And for proteins in particular, these subunits are called amino acids. So an amino acid, the name is very descriptive of the chemical functionality in that molecule. There is going to be an amine group, meaning a nitrogen-containing group on one end, and it's going to be separated by two bonds from the other end of the molecule called a carboxylic acid. And a carboxylic acid is an organic acid, meaning a carbon-based acid that is characterized by having a central carbon that's attached to two oxygens. One of the oxygens is attached via a double bond, so it's a carbonyl group, and the other oxygen is attached to the carbon via a single bond. And that oxygen is also attached to a hydrogen. So we call that a hydroxyl group, also known as an alcohol group. But when the carbonyl and the hydroxyl groups are attached to the same carbon, it's a carboxylic acid is what we call it. And it's called an acid because the proton that's attached to the hydroxyl oxygen is very acidic, meaning that it's very happy to leave its electrons behind and become an H plus ion and leave the rest of the what is now called a carboxylate ion behind with the extra electrons, and thus carrying a negative charge. So it's a carboxylic acid. An acid in general is a proton donor, and a base for the purposes of this class is going to be what we consider to be a proton acceptor. So this carboxylic acid is able to donate a proton. In between the amine group and the carboxylic acid, there's going to be an intervening carbon. We're going to call this the alpha carbon because it's alpha, or in the first position away from the amine. And that carbon is going to be attached to what we call an R group. It's some sort of side chain, and those side chains, that R group, can change. In fact, there are 20 common amino acids that make up the amino acids that are biologically relevant in proteins. It is this side chain which makes each of the amino acids unique. So, for example, when you are getting liquid aminos, maybe you guys have seen those before, seen that stuff in the store. Bragg's, for example, is one popular brand name. Pretty similar to tamari or soy sauce. It's just a broth made of liquid suspension of various amino acids plus some sodium. All right. So as I had said, there are 20 amino acids. There are more than 20 amino acids, but there are 20 common amino acids in biological systems. There are in bacterial systems. You can run across some really unusual amino acids that are very uncommon, but by and large, there's 20 that make up a human. Let's leave it at that. So these 20 amino acids can get broken up roughly into 4 categories, where the categories all share chemical similarities. So the first of those categories are hydrophobic amino acids. And these are amino acids where the side chain is made up predominantly of carbon and hydrogen. There are not really very many polar atoms in those side chains. So the simplest of the amino acids is glycine. And you can see that the side chain in glycine is just an H. And then they get more complicated. So alanine has a methyl group or a CH3 group attached to the alpha carbon. And they can get bigger and bigger. Some of them, like phenylalanine, for example, has a phenyl group. That's an aromatic hydrocarbon. So it's a six-membered ring made of carbon where the carbons have alternating double and single bonds. Tryptophan is a very important hydrophobic amino acid. Tryptophan is the starting material for neurotransmitters in the brain, such as serotonin and melatonin. Serotonin is sort of the happiness neurotransmitter, or melatonin is a neurotransmitter that regulates your circadian or sleep cycle, et cetera, et cetera. So, and a sort of diametrically opposed group of amino acids to the hydrophobic ones are the hydrophilic. So hydrophobic is water hating, fearing, phobic is fear. So those CH3, those carbon and hydrogen containing side chains, they don't want to be in water. They're not polar. They're not really easily polarizable. And because water is a polar solvent, they're not polarizable. And we know that oil and water don't mix. So these hydrophobic amino acids are kind of oily, if you want to think of it like that, and don't really like to be exposed to the water and by the aqueous environment. So hydrophilic amino acids, on the other hand, are polarizable. They have, if you look at all of those side chains, you'll notice that there are side chains that have a lot of hydroxyl groups. So serine, kind of the simplest of the hydrophilic amino acids, has a carbon, a CH2 group with a hydroxyl group attached to it. And the OH, or the alcohol, or the hydroxyl group on the serine is polarizable, and serine is happy, or at least happier, being exposed to the aqueous environment. So there's a hydroxyl and threonine. Cysteine has a thiol group, which is an SH, and that's also a polar. That's a polar chemical moiety or group on the side of cysteine. Then asparagine, glutamine have these nitrogen components, and tyrosine also has a hydroxyl group on it, so it's quite polar as well. So the other two categories of amino acids are both hydrophilic, but these carry charges. They're hydrophilic, and they're even sort of more extreme than the neutral hydrophilic amino acids. There are ones that are basic, meaning so I said a base is a proton acceptor. They have accepted a proton, and as they're drawn here, and carry a positive charge. And then there are acidic ones, and so we're showing the acidic ones. An acid is a proton donor, and we're showing them having donated their proton and thus carrying a negative charge. So on these hydrophilic basic and hydrophilic acidic amino acids, we're showing them in their conjugate forms, meaning that the base has already picked up an acid, and the acid has already donated their proton, and so that they are in their conjugate forms. Those hydrophilic charged basic amino acids are shown as what are called conjugate acids, and the charged acidic amino acids are shown as their conjugate bases. All right. So the point of all this is not to get you to memorize the structure of any of those amino acids, because that's not what I'm interested in. I want you to recognize that there are different types or different chemical categories of amino acids. And so we know that proteins are macromolecules or polymers. These amino acids are going to have to link up to one another in a chain. And the way they do that is through what's called a condensation reaction, where one end of the amino acid, the amino group, gets protonated. It acts as a base and accepts a proton, and the the carboxylic acid end acts as an acid and donates its proton, and so it has a negative charge on it as a carboxylate group. Those two opposite charges on two adjacent amino acids are going to be attracted to one another, and they go through what's called a dehydration reaction, where we essentially just pull water out and form a bond between the two adjacent amino acids. That bond is called a peptide bond. So when we look at this, what's now called a dipeptide, because it has two amino acids in it, di meaning two, that peptide bond is the C double bond O NH bond, that bond between the C and the N, the C of the carbonyl group and the NH of the amine. That bond between the two is called a peptide bond. So that's a dipeptide, right? A peptide is not a protein yet, but it's like a teeny tiny fragment of what will be a protein. A protein is a chain of amino acids that has many, many, many peptide bonds in succession. So for example, here is the sequence of a protein called cytochrome C. And you can see that, so I'm using the three-letter abbreviations for these amino acids. So GLY is glycine, aspartate, valine, glutamine, lysine, glycine, lysine, lysine, isoleucine, phenylalanine, isoleucine, methionine, et cetera, on down the line. So each of those amino acids are hooked up with a peptide bond to the next one in the chain, and it forms this long peptide chain. All right, so this is called the primary sequence of a protein. It's the order in which the amino acids are connected to one another via their peptide bonds. And I have it color-coded. The ones in green are the non-polar amino acids, and the ones in orange are the polar amino acids. And then the ionized ones, the acidic and the basic amino acids, are in red. And so this whole chain of peptides can be depicted, in this ribbon diagram. You see this picture of cytochrome C here. This is sort of the ribbon diagram of a protein, and this would be like the protein structure of an individual molecule of cytochrome C. It's just some protein that gets expressed in a cell and has some particular job. All right. Primary structure, it's just the linear sequence of all of the amino acids. So if you were to look at this linear sequence of amino acids, you'd see the side chains sticking out, those R groups sticking out from the backbone of the peptide. So here I've zoomed in, and we see a little section of the peptide. You can see alanine next to aspartic acid, and then another alanine in that chain. So each of those side chains are going to give the protein its unique characteristics. They're going to help it fold in a particular way. It's going to create active sites for like the enzymatic activity that a protein might do, etc. Whatever function the protein has in the cell is going to be conferred by the specific chemistry or chemical nature of these side groups that are sticking off the backbone. All right, on to secondary structure. So we have the linear sequence. That's the primary structure. But some proteins, in fact, most proteins, are going to have a little bit of what's called secondary structure. They don't have to, but they can. And there are two types of secondary structures. There's alpha helix. So this is basically just a spiral where that in that top picture there you see the blue arrow spiraling around, and the hydroxyl the proton from the amine in the peptide bond is able to hydrogen bond to the carbonyl oxygen from another amino acid that's like 3 amino acids down the chain from it. And so it forms this sort of tube, this coil, or a helix. We call it an alpha helix. So this is one of the two types of secondary structure. Another type is what's called a beta sheet, and this is where you have a peptide bond that forms a sort of wavy line, and then it is able to form hydrogen bonds with another part of the peptide chain somewhere else in the sequence that is going to line up adjacent to it and form this regular pattern of hydrogen bonding. So you can get these broad sheets of peptides or these sort of helical tubes. These are called secondary structural elements. So if you combine alpha helices and beta sheets, you can get all kinds of shapes of proteins. You can get long fibrous proteins. So you'd find this in like hair and silk and tendons and ligaments that hold bones together. They're like long, sort of ropey, strong building material of the body. And then there are globular proteins that, as the name would lead you to believe, are really just big globs. And they have sort of a compact, more spherical shape. And the bulk of the proteins that are able to be dissolved in water are going to be globular of some sort. Because it's minimizing surface area to volume in that globular shape and sort of hiding a lot of the hydrophobic amino acids on the inside of that protein and keeping the hydrophilic ones on the surface. There is a third category that sort of puts these two things together in what are called transmembrane proteins or membrane proteins. These are proteins that have a portion of them that acts as an anchor in the membrane of a cell. That anchor is going to have some kind of hydrophobic character to it. That is to say that there are going to be amino acids that don't want to be around water. They're the greasy side chains that we first talked about. And they're going to be very happy to line themselves up along with the greasy tails of the phospholipids, of the fats that form the membranes of our cells, the plasma membrane, the membrane that serves as the wall of any of our cells. And then there's going to be a globular part to the membrane protein, a part that is going to be exposed to the solvent environment, the water environment, and they're going to have amino acids that are hydrophilic on the surface, and then they'll probably have some kind of hydrophobic amino acids that are embedded in the center of the protein. All right, so those are the three categories, and that's called tertiary structure. Right, these sort of large tertiary structure, you can think of it as the overall shape of the protein. Right, that tertiary structure is going to be what gives the protein its ultimate functionality. Tertiary structure. So the things that give a protein, that enable a protein that is essentially just a linear chain of amino acids, the ability to fold up into the correct tertiary structure, these are interactions that are either hydrophilic or hydrophobic between the various amino acids. So you can see here we have this chain that has a sequence of hydrophobic and philic amino acids alternating. And then we can take that chain and fold it up so that all of the hydrophilic amino acids are sticking on the outside of the protein where they're exposed to water, and all the hydrophobic amino acids that don't want to be around water because they're not polar, they're all with each other, happily tucked away on the sort of greasy core of the protein. All right, so those are the forces that help a protein fold into the structure. It's important to understand what forces a protein to fold, because when we're in a little bit here, we're going to talk about cooking proteins somehow, or what's called denaturing them. That is the process of taking a raw protein and then converting it into some form that people like to consume, that tastes good to us. So to understand how that works, we have to understand the forces that we're trying to undo with the cooking or whatever it is that we're doing to the protein. So there's some other forces besides just standard hydrophobic and hydrophilic interactions with the solvent. There are these side chain interactions that can happen. We can have ion-ion interactions between oppositely charged amino acids. We know opposite charges attract. You can have hydrogen bonding interactions. So here, the second one down is two serine molecules that are hydrogen bonded to one another, and that's going to help hold the protein together, help give it its three-dimensional structure, its tertiary structure. And you can form actual covalent bonds, so that where you form what's called a disulfide bridge between two cysteine molecules. You can have the two sulfurs form an actual covalent bond. So if you look across these three, there are ionic interactions, there are hydrogen bonding interactions, and there are covalent interactions between side chains that can give a protein its overall structure. All right, so let's put it all together. The different structural elements in a protein. There was the primary structure. This is simply the sequence of the amino acids in the protein. There's the potential for secondary structural elements. This was alpha helices or beta sheets. All of this stuff can come together with a number of forces, hydrophobic and hydrophilic, and the side chain interactions to give the tertiary three-dimensional structure, or the global fold of the protein. Some proteins, and this is a bit of a nuance, some proteins, not all, but some proteins have, for their final structure, they actually have multiple peptide chains, multiple chains which interact with one another. So two or more of these polypeptide chains can come together. So for example, this is what hemoglobin is. Hemoglobin is what's called a tetramer. It has 4 globulin subunits that give the protein its overall fold. And we call that quaternary structure. That is not every protein has a quaternary structure. All right, so on to the that's all the background of what a protein is. Now let's talk about what it means to denature a protein. So denaturing a protein is taking a protein, we're going to consider the one in the middle, and this is the original protein that's active. This is the way it is the natural way that it's supposed to be folded in this cartoon. And denaturation is some sort of disruption of that native structure. And it can, a number of things can happen to a protein when you try to denature it. You can completely unfold the protein so that it's like some giant piece of spaghetti on the right, or it can slightly alter the structure, the tertiary structure of the protein, so that it gets incorrectly folded. The protein is still there with like secondary structural elements, but it doesn't have the right 3D topology. So there's different ways a protein can be denatured. When we're talking about food denaturation, there's really 5 ways that humans have of denaturing a protein. These correspond with different ways we have of preparing food, specifically protein. So I just listed them here. The first one of the things you can do to a protein is to change the solvent. So I don't know if any of you have ever had mezcal. It's a type, or seen it. It's a type of alcohol that's very similar to tequila. And often in the bottom of these bottles of mezcal is a worm. There's some sort of worm. I forget the type of worm. But the proteins in that worm have been denatured because of moving that worm from its normal aqueous environment, and you drop it in a bunch of really high concentration alcohol, and it changes the solvent. It changes those hydrophobic and hydrophilic interactions that maintain the native fold of the proteins, and it disrupts that native fold, denaturing the protein and sort of rendering the protein inactive and edible to us. I mean, it would be edible raw, but I guess it's a little bit better sitting in the bottom of a bottle of mezcal. Another way to denature proteins is to change the pH. That is to change the acidity of or basicity or alkalinity of the protein. And so here's an example of that. Maybe many of you have had ceviche before. This is taking, it can be raw fish or it can be seafood of some sort, and you just squeeze a bunch of lemon juice over it, and that lemon juice changes the pH of the fish and denatures the protein. So the fish that you would have in the ceviche may start translucent when it was raw. Then you add the lemon, and with time, the fish or the seafood becomes opaque, and that is because you are denaturing the protein. You're changing the optical properties of the protein and making it more palatable. You can also add various ionic substances. So it's very common to, particularly historically, to take meat that had been wild game that had been caught, take the meat and then salt it. And that salting process disrupts the structure of the proteins and cures the meat, so to speak, by adding that salt. The one I'm sure we're all the most familiar with is changing the temperature. So if you raise the temperature, this is what we call cooking. And cooking is a great way to denature the protein. You're adding all this thermal energy to the protein, disrupting the side chain interactions and unfolding the protein, denaturing it irreversibly. And then the last way is simply just violent agitation, messing the protein up, beating it up. So for example, this is what meringue is, or a whipped egg white. You take the albumin in the egg white and you just whip it so the protein itself gets knocked around and eventually breaks down, denatures, and is not able to fold again properly. And it forms this, goes from translucent in the egg white to this opaque color when an egg has been whipped into a peak. So we're going to look at each of these in more detail. First, let's start with the mezcal worm, changing the solvents. So if you go from a polar solvent like water to a little bit more non-polar, or the one that's a little bit more hydrophobic, like some kind of an organic solvent, that is going to slightly shift the position of the hydrophobic and hydrophilic side chains in the protein. So maybe some of those hydrophobic amino acids in the center of the protein will be happier interacting with a nonpolar solvent and will move to the surface of the protein. And hydrophilic side chains may want to cluster together on the inside. So Here on the left is some sort of idealized protein surrounded by water, and it's ordered properly. And then we put it in ethanol or alcohol, right? It's a type of alcohol. It's drinking alcohol. Ethanol is polar in the hydroxyl group and the alcohol group that's on one end of it, much like water, but it's got this big nonpolar end to the molecule. So it's sort of like an intermediate polarity molecule. It's not as hydrophilic as water. It's a little bit more hydrophobic, and it sort of leaves room for those hydrophobic amino acids on the inside of a protein to sort of emerge from the center of that protein and thus denaturing the structure of the protein. All right. So next is changing the pH. So this was making ceviche, for example. That was the example I gave you, putting lemon juice on the seafood. So if you lower the pH, lowering the pH is going to protonate everything. It's going to protonate the carboxylic acid side chains. So normally at neutral pH, or 7, for example, you have some protein in its native state where you have an aspartic acid that's attracted to a lysine, a basic amino acid, and that opposite charges of those two amino acids make those two side chains attracted to one another, stabilizing the three-dimensional structure. Well, if you lower the pH, that is, dump a bunch of protons into the solution, you are going to drive that equilibrium towards the protonation of the aspartate to aspartic acid. And once aspartic acid has been, aspartate's been protonated to aspartic acid, it's not going to be as attracted to the lysine any longer. And then that will have destabilized that side chain interaction that will have been holding the tertiary structure of the protein together. Conversely, perhaps you would have the charge repulsion between two side chain groups that are the same, that have the same charge on them. And that repulsion is part of what is stabilizing the three-dimensional structure. You dump a bunch of acid on the protein, protonating both of those, and suddenly you're able to have a hydrogen bond form, and those side chains are not as repulsed by one another as they were before, thus again disrupting the native three-dimensional structure, or tertiary structure of the protein. Okay, so that's essentially what's happening when you're changing the pH. It can go in the other direction as well. So imagine if you have, it doesn't have to be acid. It can also be adding a base, alkalizing the protein. So normally at neutral pH or pH 7, two side chains are attracted to one another of opposite charge, as we've already seen. But then if you dump a bunch of base in it, it's going to tear proteins off of the protons off of the basic amino acids. So, for example, the aspartate is not going to be as attracted to the deprotonated lysine. Again, just disrupting the three-dimensional structure of the overall protein. And it goes in the other, similar things happen when you change, when you add an ionic substance. So in this case, we're going to add salt, right? So this was like salt curing meat. We have the charge interactions that we've already described, and we disrupt those by sort of shielding the charge on the aspartate and the lysine. These are just examples of amino acids that may be interacting with one another. We shield the negative charge on the aspartate with the sodium ions, which are happy to cluster around that negative charge, and we shield the positively charged amine on the lysine group with the chloride ions that are attracted to that positive charge. And because those charges on the two amino acids have been shielded by all that salt that we've added, the side chain interaction that would be present in the native state has been disrupted, and you've destroyed or compromised or denatured the tertiary structure of that protein. All right, on to everybody's favorite way to denature protein: cooking, raising the temperature. So there's two possible things that can happen. You can either reversibly or irreversibly denature a protein. So first of all, let's just think about the more common one, that is the irreversible denaturation of a protein. You have some kind of active protein. You're going to heat it up. That first arrow represents heat. And you inactivate it, you unfold it, and you unfold it to the point where, because of that protein, because of whatever the nature of that protein, it's not really able to reconfigure itself. It's not able to find its way back to its native state. Maybe the open chain protein, the denatured protein, interacts with other denatured proteins around it to cross-link themselves, and they're not able to reform into the tertiary structure that they started with. So this is what happens in frying an egg, for example. That clear albumin protein denatures those open chains of protein, and the albumin interact with one another, cross-link and whatever, so that the protein is not able to refold itself the way it should. This is very common. Like, this is what happens to most proteins when they get denatured. You tear the protein apart, suddenly it's going to have all these potential interactions with other protein chains, peptide chains that have been denatured and torn apart, and you get cross-linking, and it's just one big protein mess. On the other hand, there are certain proteins that are able to be denatured, and then, but that's a reversible process. So if you let the milk, so an example is warming milk. You can warm milk, denature the protein, and then when you let it cool, often that protein can refold itself back into the shape that it was originally in. So you can scald milk and let it cool down, and it's reasonably the same as it was before you heated it. So there's a reversible and irreversible denaturation possible with temperature. So here is a little bit closer look at what's actually happening with this irreversible denaturation. You have the albumin from an initially cracked egg. It's all water soluble. It's translucent. We're adding heat to it, and with time, that protein becomes water insoluble. So in the cartoon on the bottom, you have all this albumin, and there are these native thiol groups in there that the heat is going to disrupt. It's going to tear the protein apart. Those SH groups that were embedded, were buried down inside the body of the albumin proteins. They get exposed and are able to interact with one another and form these disulfide cross-linked bridges, these covalent bonds between them, between peptide chains and proteins that should never have been interacting with one another. And so this like large mass of intercross links protein is what gives the egg yolk its sort of firm shape, yeah, so that it's not liquid and able to move around like it was when it was a raw egg white. And it also is what changes the optical properties of it, making it go from clear to white. So as temperature increases, the activity of a protein goes up. So the temperature is along the x-axis here, and activity is along the y-axis. If we look at the blue line, that's activity. As you raise the temperature, activity goes up, hits a peak, and then drops off quickly. And if we look at what's going on and compare this to the fraction of the proteins in a sample that get denatured, we see that activity, that the fraction of denatured proteins, it begins to go up with temperature above 40 degrees. Most proteins then get fully denatured around 60 or 80 degrees, and at which temperature you've lost all the activity. So why is the activity low at a low temperature, right? We know that body temperature is about 37 degrees Celsius. Let's go lower than that, around 20 degrees Celsius. Why is the activity of a typical protein so low? That is, when I say activity, a protein does a job, right? A protein has a job. Often they're enzymes. Enzymes are essentially just catalysts or little molecular machines that are doing some job. There's an activity, like how well they do that job. Activity is low at the low temperature because often that protein needs thermal energy, temperature energy, to do its job. And if there's not a lot of temperature there, then the activity, the ability of that machine to work is kind of reduced. But as you give it more energy, the activity goes up and up and up, but then it eventually drops down again. And that is because we begin to denature the protein, and there's not enough protein. There's like too much energy for the protein. It gets torn apart, it gets cooked, and once the protein has denatured, you lose all the biological activity that that protein might have had. So, yeah, you can affect the cooking of a protein with temperature, but also pressure. So the thing about temperature is it is in this interplay with pressure as well. You can affect the native structure of a protein not just by changing the temperature, but you can affect it by changing the pressure. So, for example, you can denature a protein at a lower temperature if you just increase the pressure that that protein is experiencing. This is what pressure cookers do. They're hot. They're not extremely hot. They're just at a really high pressure. And putting a protein under that high pressure, slamming a whole bunch of them together under high pressure, that facilitates the denaturation process. So here you see this picture, this diagram on the left-hand side. Along the x-axis is temperature, and along the y-axis is pressure. So, and you can see that as the pressure goes up, it's easier for you to get to a denatured protein. And likewise, as the temperature goes up. Yeah, this is the basic idea. All right. So when you eat a protein, your body is going to take that protein and break it up into the individual amino acids that make up that protein. And those proteins that you've consumed don't have any of their original biological activity. They get broken up into each of the little amino acids. So your body is going to make enzymes that are going to clip off the amino acids on the end of the peptide chain for all the proteins that you eat. So here's a picture of all these different foods that are dietary sources of amino acids. We get protein from nuts and seeds. They're a great source of protein. Obviously, meat has a lot of protein in it, so red meat, fish, chicken, all that kind of stuff. But then a lot of the dairy food has protein in it. So, for example, egg or cheese, milk. And then lastly, a great source of amino acids or protein comes from the legumes or beans. So we see a bunch of different kinds of beans there, lentils, split peas, etc. It's soy milk, soybeans have lots of amino acids in them. So lots of protein in it. So those are all great dietary sources of amino acids. Because the body breaks, your digestive system breaks a protein down into the individual amino acids that make them up. This is why. So insulin is a protein that your pancreas makes. The beta cells in your pancreas make insulin, which is a small protein that is responsible for helping the cells take up sugar into themselves. This is why insulin has to be injected. It can't be eaten. It is a protein, but we can't eat insulin because our body would just digest it. It would break the insulin down into the amino acids, and we need the insulin to still be biologically active. So we have to inject it into the body, bypassing the digestive system so that that protein, that insulin protein, is still able to do its job and signal to the cells that it's time to take up the sugar that's in your blood. All right. This brings us to the concept of nitrogen balance. So, when we first started talking about the structure of an amino acid, I showed you that there was an amine group on one of the three atoms along the backbone of an amino acid was a nitrogen. This is the whole reason we're using nitrogen fertilizer, et cetera, et cetera. It's getting nitrogen into the food chain to make those amino acids. You need that nitrogen. So the idea of nitrogen balance is that in a person, a typical person who doesn't have some sort of extenuating circumstance that we'll talk about in a moment, a typical person, the amount of nitrogen that comes in their body in a day should be equal to the amount of nitrogen that leaves their body in a day via either their feces or their urine. So in this model I have, this scheme that I have on the right-hand side, you see the dietary protein that's coming in the day, maybe 80 grams, something like that would be a reasonable amount for a person who's not weight training or doing anything unusual, just a normal person living their life would eat 80 grams of protein perhaps. And that dietary protein has a number of number of fates. So it can be broken up into amino acids and dipeptides. And those amino acids can enter the nutrient pool of the body that can be either made up into the proteins that make our body, and those proteins act as enzymes to do all kinds of things. But in this case, we're talking about the enzymes that are in the GI tract in your digestive system that are going to help break down your dietary proteins into the amino acids that are part of the nutrient pool. Or the amino acids in the nutrient pool can get broken down themselves into various metabolites like urea or uric acid or ammonia. These are nitrogen-containing compounds that are going to be water soluble and are filtered out of the blood into the urine. So the vast majority of the nitrogen that you consume in a day, if you're in nitrogen parity or nitrogen balance, then that nitrogen leaves the body predominantly through your urine. There is a little bit of nitrogen still in your feces, about 10 to 12% of it. This is why that number is a little bit higher in other animals. This is one of the reasons that, for example, sort of repulsively, rabbits like to eat their own feces. It's to be able to, or dogs will also eat feces. It's a way of them reclaiming that nitrogen, that protein that is in that feces, still getting a little bit, eking a little bit more out of that stuff. Humans have found other ways. All right, so this balance between nitrogen intake and nitrogen output can be disrupted. And so, for example, a person can be in positive nitrogen balance. That means your body needs to consume more protein than is nitrogen than your body is losing through the urine and feces in a day. So what are some examples of people who might be in a positive nitrogen balance? We see bodybuilders. They're putting on muscle mass, so they need more protein than they're excreting. Pregnant women, these are people who are putting on body mass as they grow the fetus, and they're absorbing more nitrogen than they're excreting. Any kind of child or person who's growing, an athlete, anyone who's putting muscle mass on, that's going to be a person who's in a positive nitrogen balance. On the other hand, a person in negative nitrogen balance is somebody who's losing nitrogen. The nitrogen that's being excreted from their body in their urine exceeds that of the nitrogen that they're absorbing through their diet. So some examples of that would be a person with anorexia nervosa that you see there in the first diagram. This is a person who is losing nitrogen, losing protein. Their body is breaking proteins down because they're not eating enough. A person who is suffering from malnutrition because they just cannot get enough food in their body because of whatever condition they're living under. And a person who has some sort of muscle wasting disorder. So, for example, a person like Stephen Hawking there, who is losing muscle mass because he has Lou Gehrig's disease. This is a person in negative nitrogen balance. It doesn't have to be anything as extreme as either of these three, too. It could just be Arnold Schwarzenegger, that bodybuilder in the upper corner there. It could be him after he has stopped bodybuilding and he's losing muscle mass. He would then go into negative nitrogen balance at that point. Okay, we're going to stop it there and we'll pick up next time.